So my name's Aaron David, and I'm pleased to introduce today's speaker, Avery Brooks. Avery is from Central Florida and graduated from Stetson University in 2025 with a BS in biology and environmental science. As an undergraduate, she conducted research on arbuscular mycorrhizal fungi as part of sandhill restoration, an experience that really served as excellent training for the project that you're going to hear about today. She also spent some time as a museum guide at the Gillespie Museum in DeLand and interned for the Institute for Water and Environmental Resilience and the Stetson Campus Climate Environmental Explorer. Avery's been a wonderful team member in the plant ecology program. She's a super quick learner and an absolute joy to have in the lab and the field. And on top of that, Avery's project is super cool. So as many of you know, our oak scrub actually has quite a large component of heath plants in the plant family Ericaceae. So these are the feather bushes, blueberries, huckleberry, roseberry, Florida roseberry, to name a few. And these heath plants actually have their own unique mycorrhizal fungi associations known as the Ericoid mycorrhizal fungi. So they're thought to be mutualistic, but really virtually nothing is known about these fungi at all. And so they are in effect the forgotten fungi. So despite the thousands of acres of ericaceous plants in the Florida scrub, to my knowledge, no one has checked to see if they associate with these relatively unexplored fungi until today. So I don't want to hyper talk up too much.
The high bar.
Discovering new microscopic mycorrhizal fungal structures, you are in for a treat. So after our internship, Avery is headed north to North Carolina and will be working with the North Carolina Conservation Corps. And with that, I'll turn it over to you, Avery.
Hey everybody, as I said, my name's Avery Brooks, and I'm a plant ecology intern funded by the Von Jordan Foundation. And today I'm going to take you beneath the ground into the soils to look at these special little fungi that make these networks with plants and even in the soil itself. So let's get into it. Before we get into mycorrhizal fungi, let's start with fungal diversity. There are many unknowns in the world of fungi as their diversity is extremely vast. In fact, there's an estimated 12 million species of fungi where only about 155,000 of those have been currently named and identified. With only about 1% of these fungi having been named so far, there's a lot of potential for new discoveries along the fungal tree of life. But where can we look for these new fungi? Well, rare habitats can house a wide variety of new species. In fact, a lot of studies looking at rare habitats with these unusual niches have led to the discovery of many new species where they have found them growing on these unique substrates not found in other areas. One such rare habitat is the scrub. And there is limited fungal research so far in the scrub, which makes it a great habitat we can look at for these new species. The scrub is a great place to look because it has a lot of nutrient and water stress and also very acidic soils, making it maybe in need of more mutualisms. And also there's a lot of endemic plants there, leading to maybe more special species. These factors could enable a lot of unique fungal species to emerge. So specifically, we can look towards roots for fungi. When looking at fungal diversity, it expands beyond forms like mushrooms and to yeasts and molds, and the focus of my project, root-associated mycorrhizal fungi. These mycorrhizal fungi are often microscopic, forming these web-like mycorrhizal networks in the soil and symbiotic associations with plants. These associations are really the driver of a lot of nutrient cycling in ecosystems, as the fungus exchanges nutrients with the plant in this nutrient cycle. The fungus will often give the plant nitrogen or phosphorus, and in exchange, the plant will give the fungus carbon. In fact, more than 5 billion tons of carbon are cycled annually through these sort of processes. Therefore, studying these fungus and these dynamics of plants is important to understand how we can improve overall plant health and ecosystem health as a whole. But we barely have a grasp on what species are out there, let alone how and why these associations form. My study seeks to fill in the gaps in understanding two different types of mycorrhizal fungi, arbuscular mycorrhizal fungi and Ericoid mycorrhizal fungi, and as well as look into the scrub ecosystem. So speaking of, here is a scrub ecosystem. And these habitats are often dominated by oaks, palmettos, and heaths. And often you might hear that they're called like oak scrubs. But I've started to think of it more as a heathland in a way because it is dominated by a lot of, it has a lot of heath species. Two examples of this heath or Ericaceae family are Baharia racemosa or tar flower and Lionia fruticosa or featherbush or staggerbush. So these species you might see around the scrub, and there are many more. But we can look towards the Ericaceae family to explore fungal associations in the scrub. Ericaceous plants generally associate with two main types of fungi, arbuscular mycorrhizal fungi and Ericoid mycorrhizal fungi. AMF, as we call it, is well studied. It has, as we know, a single fungal lineage, and it associates with over 80% of plant families, so hence why it is often so well studied. It also forms these arbuscule structures within the plant root cells themselves, as it is microscopic. As you can see here, they're circled in red is the arbuscules, and that is the site of nutrient exchange with the plant. It also forms these hyphal networks where you can see connected to the spore on the diagram are all of these hyphae, and that is connecting all of the parts of the fungus. And studies have already shown that AMF is present in the scrub, specifically in the soils with DNA analysis. So I'm hoping to build on that with this project. BRM, on the other hand, as Erin has said, it's virtually nothing is known about them. They're there's a lot of new studies coming out, but what we do know so far is that there are several, it occupies several fungal lineages, and it only forms a structure within the Ericaceae plant family. And it forms these coil-like structures, which are their form are arbuscules. And they are the site of nutrient exchange within the Ericaceae family plant. And we don't know for sure if it forms a hyphal network or not. There's a lot of new studies, hopefully that will reveal that. But there's a lot of potential to increase what we do know about it by looking towards the scrub, since there have been no studies in the scrub on this type of fungi. And there's a lot of potential to look into it since we have so many ericaceous species. Plants and fungi are releasing these chemical compounds to stimulate the growth of plant roots and the fungal spores and hyphae as conditions are changing in the soil. Notably, stressful conditions can cause an increase in mutualisms that will improve the survival of the plant and the fungus. Selective pressures from things such as competition and water and nutrient stress, for instance, may influence the plants to increase the number of associations they form. And we can look towards that to see how stress impacts symbiotes. Scrub ecosystems have three main habitat distinctions. We have flatwoods, scrubby flatwoods, and rosemary scrub. And these different habitat types follow what we call an elevation stress gradient, meaning that as we get higher in elevation, we will see more stress in the ecosystem as nutrients and water becomes less available. The scrub is an ideal location to study the effects of stress on the environment, as even like minimal elevation changes have strong impacts on nutrient and water availability. As you see here, flatwoods are the considered lower stress since they have more nutrients and water available. And as we move up the elevation stress gradient, rosemary scrub is the high stress ecosystem. And the scrub, although it even has just a minimal elevation difference of only 1 to 3 meters, we see a lot of change in nutrient and water availability, making it ideal for this sort of study. And for instance, one study looking at oak species in the scrub found that ectomycorrhizal associations, another types of mycorrhizal fungi, increased with higher stress. Therefore, we wanted to continue that with other species in this study. And by looking at these impacts on these other species, we can understand how these dynamics work in the scrub. So that leads us to our research questions. The first being, which ericaceous plant species do AMF and ERM associate with? Since ERM, or Ericoid mycorrhizal fungi, have been researched in other heathlands before with acidic soils and low nutrient availability, I wanted to take that to the scrub because that is a similar system that we have here. And I also wanted to look at AMF since we've had some initial studies done, but I wanted to go more in depth on the effects of stress on AMF. Also, we wanted to look at whether or not the AMF and ERM colonization would increase under higher stress. Would these mutualisms occur more often under more stressful conditions? So first, let's look at our study species. In my study, I focused on 7 out of the 8 key species we find in our Florida scrub: Beharia racemosa, or tar flower, Lionia fruticosa, or staggerbush, Lionia lucida, or featherbush, Gay-Lusacia dumosa, or huckleberry, Vaccinium darwii, and Vaccinium marcinites, types of blueberries, and Serradiola ericoides, or Florida rosemary. And that the Ericaceae family, it's important to note that not only does it have ecological importance in our scrub, but it also has agricultural importance as it involves plants such as blueberries and huckleberries and cranberries that we eat. So it's important to see how it works, not just for agricultural, or not just for ecological growth, but agricultural growth. Now let's get into my methodology. So I was focused on 4 distinct sites that I collected a total of 150 samples from. Each of these sites was selected to have not have been burned within 10 years. This would be to ensure there was mature plant growth and also that diversity wouldn't be impacted by burning. And then for each of these 4 different sites, I was focused on the 3 habitat types mentioned before. For 6 of the 7 species I was looking at, I looked at flatwoods. I collected from flatwoods and scrubby flatwoods. I did this because I also was, for the rosemary scrub, I just wanted to focus on where rosemary would be found, which is in the rosemary scrub. And the flatwoods in this case would be the lower stress environment, and scrubby flatwoods would be the higher stress for these 6 species. For the rosemary scrub, I took elevation data as well to compare between lower elevation or less stress rosemary scrub and higher elevation, high stress rosemary scrub. For each of my plants, I took a sample of both fine roots and the thicker main roots, and this would be to ensure that I had results reflective of the entire root profile of the plant. And then I took these roots, chopped them up, and threw them into histocassettes. And these histocassettes would be to ensure that I could stain the plant roots. So what does the staining process look like? Well, I stained these plant roots to be able to see different structures of the fungus within the roots themselves. To be able to do this, I first cleared the roots, meaning I took out like tannins and other compounds through boiling them in potassium hydroxide. So once the roots were cleared, then I briefly acidified them in hydrochloric acid. This would be to ensure that the stain would stick to the roots. And then I stain the roots using a trypan lactoglycerol stain, also known as trypan blue. And this stain works by attaching to the chitin in the fungal cell walls, so that way you can see the fungal structures within the cleared plant roots. Then I mounted each of these root segments onto slides to be able to look at under a microscope. So this is what a stained structure would look like inside of the plant roots. I made slides for each of the plants and lined the roots up like so to be able to see them. To collect my data, I used what's called the point intercept method, which basically means as I'm looking at my slides under a microscope, I create an imaginary line down the center of my vision, and that line will be the one I'm staring at and looking at to see what intercepts that line. So I'm looking to see if root pieces are intersecting the line, and also if different fungal structures are intersect the line so that I can count them. AMF, to look for AMF, I looked for the number of roots that contain spore structures, hyphae structures, vesicle structures, and arbuscle structures. And what this looks like is, say I have my plant root here. This structure here is called a spore. This is a large round structure that connects to the hyphae. This is what it looks like when it is stained. Here are the hyphae that are branching between each of the structures throughout the cells and also out of the cells. Here are vesicles, which are smaller structures than the spores and tend to be more oval shaped. And then here are our vesicles, which are the branching site of nutrient exchange in the plant. To look for Ericoid mycorrhizal fungi, I looked at the number of roots that contained the coiling structure mentioned before. I didn't have many diagrams to go off of, but I was just generally looking for coiling based off a lot of these diagrams that I saw in papers. So looking at a quick example, say this is my stained plant root. This is what they would look like. And here there's a lot of colonization of AMF. I draw my imaginary line down the center of my vision, and each time I'll count, oh, this is one root that I'm running across. I also see lots of hyphae going over the line, so I would count that as one, yes, hyphae is present in this root. And then also vesicle structures, which I would say, yes, one for the vesicle is present, add one. And then some arbuscle structures, which I would also count that as well. And that's what I would do for each of the root segments I came across. Here's me hard at work, looking under a microscope for many hours. And now that leads us to our first question. Is Ericoid mycorrhizal fungi present? Well, I would say there was a high potential because we found a lot of coiling in some of these plant cells. So very exciting. Hopefully we will get more insight onto that with some DNA analysis that will be done. But this distinct coiling separates it from the AMF, and we found quite a bit, which is great. So we found this coiling behavior in 5 of the 7 Ericaceae family plants that we saw. On the x-axis, you can see the species listed there, and then the y-axis shows the mean number of coils we found in our plant species, ranging from 0 to 2% of colonization in each of these plants. But there was no significant difference between each of the plants. Next, we looked at whether or not AMF was present. And you could say that there was quite a bit of AMF present throughout pretty much all of my samples. So, which was great. I found tons of hyphae. As you can see, all of these webbing structures are hyphae. An arbuscles up there with the branching. There's some vesicles, some spores. I found a bit of all of the structures, which is very exciting. I want to draw your attention attention to hyphae and arbuscles as the main structures we're going to look at today. So hyphae I chose because it is the most abundant AMF structure that I would see, and it would give us a good look at colonization. And then also arbuscles because that is the distinguishing feature of AMF, and it is also, and we wanted to compare between species on that. So we didn't find any significance between species for any of the AMF. As you can see here, hyphae is on the left, comparing between species, and then arbuscle abundance is on the right, comparing between species as well. And we didn't find any significant difference in abundance for either. But as we explored the data, we also became interested in seeing how AMF and ERM were interacting with each other. So we decided to run a correlation test on the presence of coiling, which would be indicative of ERM colonization, and hyphae, which is indicative of AMF colonization. And we found excitedly that AMF presence is correlated with when, yes, coils are present, there are more hyphae present as well, which at least shows us that there is no evidence of them impeding on each other's growth. And it could maybe lead us to future insights that we could explore on how they interact with each other in the ecosystem and potentially benefit each other. So as I was thinking, as we look at the stress question on whether or not stress will impact the system, many studies have shown that higher stress has led to higher colonization. So we hypothesized that AMF and ERM colonization would increase with higher stress. But we didn't find any evidence of this. Looking at AMF, where species are listed below and the mean hyphal abundance is on the y-axis, we can see that there was no significance between both species before and also colonization of hyphae, indicative of AMF colonization in the habitats. So F represents the flatwoods or lower stress habitat, and SF represents scrubby flatwoods or higher stress. And so what we would see if there was a direct impact of higher associations with stress is that the blue scrubby flatwoods bar would be higher than the red flatwoods bar, and that would indicate that there are more mutualisms. But we did not see that with AMF or with the ERM, so not with the Ericoid as well. So we did not find evidence for this hypothesis. Luckily, these studies will be continued as two researchers excitingly took interest in my study and are going to be continuing this awesome work. Dr. Gaurav Kamlikar at LSU has been sent some soil and root samples from Ericoid family plants and also soil samples near Ericoid family plants. And with this, he'll be able to run DNA analysis and really tell us what fungi are there. And Dr. Ning Zhang at Rutgers, she has also been sent some root samples. And with this, she'll be able to actually culture on discs some of these fungi and hopefully describe some new taxa found in the scrub. And she hopefully will be able to culture some of these coiling structures that we see and tell us, is it Ericoid, is it not? Hopefully the answer is yes. So to wrap it up, this is the first study to have found evidence of ERM structures in the Florida scrub. And we found ERM being present in the same places AMF are, which is very exciting. But we don't really know yet what their role is in these ecosystems. Is there more of a mutualistic association, or they could also be more like commensals or parasitic even to the plant? So it's really important we further uncover more of these relationships. Also, we found colonization was not affected by environmental stress, surprisingly. this contrasts with a lot of the work that I've read and even we've seen with the previous study in the scrub. But there could be many potential explanations from a lot of abiotic and biotic factors. And one explanation could be, instead of a change in abundance we could be seeing a change in fungal diversity that we actually can't see with this data. Another study I looked at how fungal diversity would change with stress. So more stress meaning more diversity. So it's just another piece of the puzzle below ground, and showing us how complex they really are and how far we have left to go with these studies. And the Ericoid family and scrub are exciting new potential sources for fungal diversity. And as we look for what's beneath the surface of the scrub, we'll learn more about these complex ecological communities and what's really driving scrub life. So with that, I'd like to acknowledge everybody at the plant lab for dealing with me and also helping me out along the way with so much support. The Von Jordan Foundation for funding me, Matt Smith for his fungal expertise, the ranch for letting me commandeer their lab and be an evil chemist for a day, and Dr. Ning Zhang and Ken Lakar for taking on the next steps of this project. Hopefully we find some exciting new stuff. Laura Reed for running it in the back. Thank you very much. Dustin for some amazing pictures of me out in the scrub. How did I look cool digging in the dirt? I don't know. And all other Archbold staff and interns that have helped me out along the way. Here are my references, and now I open it up to questions. Yes, Abby.
If I recall it, there didn't seem to be a whole lot of association with the rosemary with regards to mycorrhizae. Do you think that's because of the chemicals they put out, or is that just something we don't know?
That's something we don't know, and it would be really interesting to see because there were some cases where I saw little presence of certain structures, and then others where I saw a lot of hyphae in the rosemary. So yeah, looking into those like allelopathic chemicals and their influence on these different fungal structures would be really great. I was wondering that too. Yes.
I have an online question from Margaret. Margaret says, great job. This project is very interesting. Mm-hmm. How did you decide on the statistical analyses you described in this presentation? Also, do you have any ideas for your next experiment yet?
Oh man. Yeah, so I decided on them just looking at the factors of, well, I was running ANOVAs and I was comparing between the factors of habitat and species. So running like binomial tests on those. And I, for next steps, I really would love to do some more fungal research for sure, looking at new fungal species. Mycorrhizal fungi have my heart, I think, after a lot of this research and previous research. So maybe looking into some forest ecosystems would be really great. I don't know. See where it leads me.
Yeah. Do you have any idea of what the effects of fire would be? Just curious if when you sampled, if you held fire frequency constant for time since fire.
Yeah, so I took out that variable. That was in my original plan, but I took that out just so I could make sure I could focus on the Ericoids. But I am really interested to see if there's any impacts on burning. Some studies have found there have been a little bit of impacts, mainly not. Mainly they've seen that the below ground structures seem to be the same with fungal diversity and abundance. It seems like they're almost exempt from fire in some cases, but I'm sure if as we look into it more, we could find out even more about that. Any other questions?
So not really a question. First of all, you did a terrific job.
Oh, thank you.
So a couple of observations. One is at Rutgers, I suspect McGinn's predecessor, John Dighton, she came here quite a few times.
Oh, really?
And he was interested in thinking about Ericoid. And there was some work from the New Jersey Pine Barrens that
was architecture, actually.
oh, it was.
Yeah.
Okay. So shut up. But he did do a couple of publications on Ericoid, mycorrhizae there. I think they were mostly descriptive, but I just wanted to give
Oh,
it's the closest I can think that we would have for a similar system. And then the other thing I would throw out is, you know, we always think about drought as a stress here, but under high water conditions, water is a stress here, at which point those flatwoods can be under, at a completely different time, a completely level of stress. So just saying that the higher elevations are always the high stress environments may not be, it may be a bit of a simplification. It may be depending on the groundwater level at the time. Anyway.
No, definitely. And in this case, I was really looking at areas that were not like saturated or inundated with water to eliminate that sort of factor. I was mainly focused on, the upward stress gradient, but you're totally right. There can be a stress in the opposite direction as well. And so I eliminated that water stress factor by making sure none of my samples were really saturated. Yes.
I mean, kind of in that vein of thought, do you think that seasonality could affect the abundance of these mycorrhizae?
Yeah, 100%. Especially, we've seen a lot of fungal growth explode after heavy rainstorms. So, and they can persist, but even with drier conditions. But really, you see a lot of fungal growth with wetter conditions, and that would be another great study to look more into that under wet and dry conditions as well.
Can I ask a follow-up to that? Is that sort of half explained by the spores? Do they tolerate those drought conditions in sort of little coated spore structures?
Yes, they're hardier structure. The growth kind of just halts a bit without the presence of a lot of water. But once it starts getting hydrated again, it'll start growing more rapidly. Yes.
What did you say that the fire cutoff was in terms of how many years since?
I made sure there was at least 10 years since the last burn.
Do you think that that would have introduced additional stress to the flatwoods plant, given the expected shorter fire interval of those ecosystems?
That's a good point. It definitely could have been that too, and that could have been another factor, another abiotic or biotic factor on the ecosystem.
Because I don't know if that would be a
I didn't necessarily think
why you would have the lack of, you know, the association that you would expect with
we
fire
didn't
stress.
see the changes. And that could have been, and then looking at maybe burned systems would be a next step. That would be really exciting. That could have been an influence. Yes.
This one is from Jade online.
Okay.
Do you know about the geographic range of ERM? I wonder if it can be found in, for example, pine rockland type habitats that have Lyonia species.
I did find one study on pine rocklands and a few on woods and also some in heathlands, especially in Australia and New England. So yes, so they definitely have wide dispersed. They're widely dispersed into some of the northern areas. And I did find one study on the pine rocklands as well. So.
Last question. All right, well, let's give her another round.